So, a very good afternoon to all. Today I will be talking about genetic disease. So, a genetic disease, it occurs when a person has one or more abnormal genes or a missing genes, extra genes, inactivated genes, or overly active genes that can lead to a medical condition. So first, before going into the genetic diseases, let me just give you a small brief about genetics. So what is genetics all about? So first, what is a gene? Okay, so genes are the building blocks of heredity that are passed from the parent to the child. These genes are found within the cells of all the organisms, and individuals' genes are present in a large molecule which is called as deoxyribonucleic acid, or is called it as DNA. So you can see here, this is a gene. Okay, so genes is a sequence of DNA or RNA that codes for a molecule that has a function. The transmission of genes to an organism's offspring is the basis of the inheritance of phenotypic traits. So these are the genes, okay, and they are a sequence of DNA. And here there's a picture of DNA, where the full form is deoxyribonucleic acid. It is a molecule which is composed of two chains which coils around each other to form a double helix carrying the genetic instructions used in the growth, development, functioning of all the living organisms. So here we have, these are the two chains, and inside the two chains we have the bases pair. And these bases pair are known as guanine, cytosine, thymine, and adenine. So these genes, they hold the DNA, the instructions for making the proteins. So basically the function of genes is it holds the DNA and they provide instructions for making proteins. What are proteins? Proteins do most of the work in the cells. They move molecules from one place to another, build structures, break down toxins, and do many other maintenance jobs. So that was about genes. C is for cytosine and G is for guanine. Okay, so you can see here the structures. This is the original sequence, whereas after mutation or after any UV radiations, so there is changes in the gene structure. Okay, so there are changes, and you can see here, this is the changed nucleic acid, or any changes in the structure of the nucleic acids. Next, coming to heredity of genetic disorders. A person's DNA is contained inside two sets of chromosomes. Each parent provides one set of 23 chromosomes to his or her offspring. Therefore, each person has 23 pairs of chromosomes, okay, which is a total of 46 chromosomes. So the X and Y chromosome, as you can see here, the X and Y chromosomes, they are called the sex chromosomes because they distinguish males from females. Now females will have a pair of X chromosome, that is XX, which while males will have X chromosome and Y chromosome. So certain genetic diseases occur when there are genetic mutations or genetic changes in sex chromosomes, and these can affect males and females differently. So if there's any genetic mutation in the sex chromosome, that is in the X or Y chromosome, then that can cause genetic conditions or genetic defects. Now, genetic conditions are usually inherited, meaning that they are passed down from parents to their children. So majority of the genetic conditions, they are usually transmitted from their parents or their grandparents. Okay, so it is generation to generation. So I hope you have understood what how many chromosomes are there and how many pairs of chromosomes are there in a human body, and the X chromosomes, X and Y chromosomes, they are called the sex chromosomes, and in a female there are X and X chromosome, whereas in a male there is an Y chromosome. Next, coming to gene mutation and type. So like I've taught you, gene mutation is nothing but changes or errors in the structure or characteristics of the genes. A gene mutation is a permanent alteration in the DNA sequence that makes up a gene. So there are two types of gene mutation. One is acquired mutation and next is hereditary mutation. Acquired comes from the word getting from somewhere, okay, basically like a recent transmission, or getting the or there may be triggering factors which can cause changes in the genes. So, and hereditary meaning transmission from parents to children or from the grandparents to the grandchildren. So that is generation to generation. So as you can see here, this is a picture: hereditary mutation and acquired mutation. Hereditary mutation, like I said, it is generation to generation. Okay, so here in hereditary mutation, this is how it is being developed. So here is an egg and there is a sperm. Okay, so when an egg and sperm, there is a fusion between the two. Okay, and if there's any mutation occurs, for example, if there's any radiation or any triggering factors, okay, so that, or if there's any infection, so there will be error or changes in the gene structure. So that with the mutation, the fertilized egg will have a mutation. So as you can see here, there's a red color, that is the mutation or the changed structure of the DNA. Okay, now as you can see here, this fertilized egg, okay, now this fertilized egg will further be divided into this reproductive, okay, the reproductive system also will have a mutated gene. In the bones also there will be a mutated gene. In the pancreas also there will be a mutated gene. The brain also there will be a mutated gene. So it will affect the body cells of the offspring. So this is nothing but hereditary mutation. Okay, next coming to acquired mutation. Acquired meaning coming from somewhere. Okay, so this is, suppose this is a normal bone cell. As you can see here, there is no mutation. Okay, there is no red color in between. So this is a normal bone cell. Okay, so if there's any mutation occurs, for example, the person is being exposed to an radiation. Okay, so after being exposed to a radiation, there will be a mutated, so the genes will be altered. As you can see here, this is another structure here, altered gene. Okay, so after the mutation occurs, the gene will have a changed, there will be a change in the characteristics and structure. Okay, then this altered gene will further be divided into altered bone cells. Okay, and here you can see in another portion, the normal bone cell. Okay, if there is no mutation, then it will further it will further be developed as normal bone cells. So this is a very simple diagram to differentiate between a hereditary mutation and an acquired mutation. Clear? Go on into a hereditary mutation. So what is hereditary mutation all about? Okay, like I said, hereditary means transmission from parents to children or from grandparents to the parents or from generation to generation. Hereditary mutations are inherited from a parent and are present throughout a person's life in virtually every cell in the body. Okay, so like I said, if there's any error coming from the parents, okay, or any mutated gene coming from the parents, the offspring will have mutated gene in all the body cells. Can be in their reproductive system or in the brain, or there can be any mutated gene in the pancreas or bones or lungs or liver or anything. So that is about the hereditary mutation. So these mutations are also called as germline mutations because they are present in the parent's egg or sperm cells, which are also called as germ cells. So hereditary mutation is also known as germline mutations. Why? Because they are present in the parent's egg or sperm cells, okay, which are also known as germ cells. Now, when an egg and a sperm unite, okay, when an egg and a sperm unite, the resulting fertilized egg cell receives DNA from both parents. So, like I said, the fertilized egg will have the will be getting the DNA from the mother as well as from the father. And if there's any mutated gene occurs in between, so the offspring will have a mutated gene in the body cells. If this DNA has a mutation, the child that grows from the fertilized egg will have the mutation in each of his or her cells or any body cells. Okay, so I hope that is clear about hereditary mutation. This is also another picture. As you can see, affected father. Okay, these are the so you can see here the pink color is the affected father and unaffected mother comes in a light pink, or you can see as in a brown color. Okay, so when the fusion of the egg and sperm will take place between the mother and father, okay, so the offsprings will have fertilized, they will have a mutated gene. So let us see here that we can see here this is the first child, okay, this will be a affected child, okay, then the second will be an unaffected child, then the third will be unaffected child, and the fourth may be an affected child. Okay, so we have you have learned in 12th science about the dominant and recessive chromosomes or the transmissions. Okay, so that applies in this hereditary mutation. Now coming to acquired mutation. Acquired mutations occur at some time during a person's life and are present only in the certain cells, not in every cell in the body. These changes can be caused by environmental factors such as ultraviolet radiation from the sun, or can occur if a mistake is made as DNA copies itself during cell division. So acquired mutation can occur because of any environmental factors which can cause changes in the DNA, or it can be because of mistakes when the DNA copies itself during cell division. Acquired mutation in somatic cells, that is cells other than the sperm and egg cells, they cannot be passed on to the next generation. Okay, so if there's any mutations in the somatic cells are nothing but cells apart from the sperm and the egg cells. So if there's any changes in the somatic cells, that cannot be passed, it will not be passed to the next generation. So as you can see here, okay, this is a normal bone cell. Okay, if there's any mutation occurs, suppose if this if the person is being exposed to ultraviolet radiations, then there will be altered genes. Okay, the radiation will have an impact on the genes structures, okay, and there will be an altered gene. This altered gene will further divide into two other altered bone cells. Okay, so you can see here the muted gene in the bone cells, whereas in the other structure here, this is a normal bone cell, there is no mutation in the other side, so it will further develop normally. So what are the acquired causes, or what are the causes for acquired mutation? Okay, so they can be because of drugs or radiation or viruses. Drugs may cause alkylation or oxidation, etc. Ionizing radiation causes breaks and cross-linking, and viruses may cause degradation of certain genes. So basically these are the causes of acquired mutation. Next, the types of genetic disorders. Single gene disorders. Okay, first is a single gene disorders where a mutation affects only one gene. So if only one gene is being affected, or if there's any changes in the only one gene, then that is called single gene disorders. And a very good example of this is sickle cell anemia. Next is chromosomal disorders where chromosomes are missing or changed. Okay, so very simple, that is if there's any changes in the chromosomes. Okay, for example, if there's a missing chromosome, okay, instead of being 23 chromosomes, there are like 22 or 21 chromosomes, so that is called chromosome disorders. So chromosomes are the structures that hold our genes. Okay, and a very good example of chromosomal disorders is Down syndrome. Next is complex disorders. Complex disorders is when there are mutations in two or more genes. Okay, so if there's any changes or any errors in two or more genes, then that is called complex disorders. And often lifestyle and environment play a very important role in complex disorders. A very good example of this is colon cancer. Okay, so these are the three types of genetic disorders. One, if there's a change or mutation in one single gene, that is called single gene disorder. If there is any change or mutation in chromosomes, okay, if the number of chromosomes are less, or there's too many number of chromosomes, then that also we call it as chromosomal disorders. And third is complex disorders when there are mutations in two or more genes. Next, diagnosing genetic disorder. There are genetic tests are tests on blood and other tissues to find out genetic disorders. The purposes of genetic disorders are: number one, finding genetic disease in unborn babies. Number two, finding out if people carry a gene for a disease and might pass it on to their children. Third, the purpose is screening embryos for diseases. Fourth, testing for genetic disease in adults before they cause symptoms. Then making a diagnosis in a person who has disease symptoms. And last, figuring out the type or dose of a medicine that is best for a certain person. So these are the purposes why we do a genetic test. Next, physical examination. Physical examination is very important for diagnosing evaluation of genetic diseases. Okay, so certain physical characteristics such as distinctive facial features can suggest the diagnosis of a genetic disorder. A geneticist will do a thorough physical examination that may include measurements such as distance around the head, that is called the head circumference. They will also check the distance between the eyes and the length of the arms and legs. Next, depending upon the situation, specialized examinations such as nervous system or neurological examination we call it, or ophthalmologic examinations may be performed. The doctor may also use imaging studies such as X-rays, CT scan, MRI to see the structures inside the body. Next, a personal medical history, which is also very important apart from the physical examination. Okay, in personal medical history, we have to get the information about the individual's health. Okay, how he is living his life, what are his lifestyle. Often going back to birth can provide clues to genetic diagnosis. So, what are the medical history, or what are the physical examination of a person at birth? All those medical history can give us some clues for diagnosing a genetic disease. Next, a personal medical history includes past health issues or history about his past, whether it can be his medical history or can be surgical history, hospitalizations and surgeries, or if there's any history of allergies, medication, what medications he has been taken, and the results of any medical or genetic testing that has already been done. So these are the very important points that we have to remember while taking a personal medical history. Next, family medical history. So we all know that genetic disease is a hereditary disease also. So that's why the family history is very important. Because genetic conditions often run in families, information about the health of a family members can be a critical tool for diagnosing these disorders. A doctor or genetic counselor will ask about health conditions in an individual's parents, siblings, children, and possibly more distant relatives. This information can give clues about the diagnosis and inheritance pattern of a genetic condition of a family. So, health information about each and every member of the family is very important for diagnosing a genetic disease condition. Next, what are the lab tests that we can perform apart from ultrasound, MRI, CT scan? So these are the methods used for genetic testing. Number one, molecular genetic test, or we call it as gene test. It is a study, it studies single genes or short lengths of DNA to identify variation or mutation that leads to the genetic disorder. So this molecular genetic test, what does it do? It studies the single genes, or we can say, or it studies about the lengths of the DNA to identify if there's any changed or variations or mutations that can lead to genetic disorder. Okay, so as you can see here, this is a molecular genetic testing. Here the second diagram. So you can see here, if this, you can see this is a this molecule is the length of this molecule and the length of this molecule. This looks much longer than this. Okay, so we can see if there's any changes. The next is chromosomal genetic test. Chromosomal genetic test, it analyzes whole chromosomes or long lengths of DNA to see if there's any change in the genetic. For example, an extra copy, if there's any extra copy of chromosome that can cause a genetic condition. So as you can see in this picture, this is a chromosome cytogenetic. Okay, so a mutation is absent in this chromosome, whereas if there's any mutation or any error is present, then this is the structure of a chromosome. Okay, so you can see the difference between the two. Next is biochemical genetic test. Biochemical genetic test, it studies the amount or activity level of proteins. Abnormalities in either can indicate changes to the DNA that results in a genetic disorder. So biochemical genetics is basically, it studies about the amount or the activity level of the proteins. So this is a mutation. So this is a gene which has no with no errors or no changes. And this picture here, this is a change in the proteins or the amount or the level of proteins. Okay, so now we will be dealing about prenatal diagnosis. Prenatal diagnosis will be able to detect the presence of a characteristic abnormality in the fetal development through ultrasound. So by using an ultrasound, you will be able to detect the presence of any abnormality in the fetal development. Next, it will detect the presence of characteristic substances by using a procedure called amniocentesis, where it is an invasive procedure which involves inserting probes or needles into the uterus, and a small amount of amniotic fluid will be taken out. Prenatal genetic testing. So there are 5 types of prenatal genetic test which can be used for screening the risk of the baby having a genetic disorder or for diagnosing such abnormality. So for screening, it evaluates the risk of genetic problems. It is a non-invasive procedure, and it offers to all women of any in any week of pregnancy. So there are 3 tests, that is maternal blood serum. We will take the blood of the mother and we will check if there's any other problems like disease condition or infections, bacterial infection or viral infections or anything. Next is non-invasive prenatal testing, NIPT, which I will be dealing in the next slide. Then we will have a prenatal ultrasound. So ultrasound is also very important for screening the genetic disease condition. Next, for diagnosing, it confirms or rules out the genetic problems. It gives a positive response whether the fetus is having a genetic problem or not. It is invasive and it offers after abnormal screening test or known risk factors. So first we will be having screen test and then the diagnostic test will be performed. So for diagnosing, it consists of two tests, that is chorionic release sampling, okay, and amniocentesis. So here, this is an non-invasive prenatal testing (NIPT). It is a prenatal screen testing that can be performed beginning around the 10th week of pregnancy. This blood test, is performed 10th week of pregnancy. So a blood sample from the mother is being taken, and as you can see in the picture, you see a fetus. This is the placenta, okay, and in the placenta there will be placental fetal DNA, which will be seen by this blue color DNA, and this purple color DNA, this is the maternal DNA, this is of the mother. So small fragments of cell-free DNA from the placenta, okay, it enters the mother's bloodstream. So cell-free DNA in a sample of the mother's blood is analyzed for evidence of extra or missing fetal DNA segments. So this test is basically done to check if there's any extra DNA segment or if there's any missing DNA in the fetus blood. Next, this is amniocentesis. Amniocentesis, it is an invasive procedure where a small amount of amniotic fluid is being taken out from the placenta, and then a needle is called transabdominal needle is being inserted, okay, into the uterus, and then a sample of it is being taken out, and then this is done usually after 16 weeks of pregnancy. This amniocentesis test is used to diagnose neural tube defects and genetic disorders such as Down syndrome. What are the risks for doing this test? There can be a fetal loss 1/200 to 1/300. Chorioamnionitis, that is infection, there can be chances of infection, and there will be chances of fetal injury, that is injury to the fetus. Alloimmunization or ROM. Next, replaced with quad screen measures the maternal proteins and cell-free DNA that detects the fetal DNA in mom's circulation. So this is amniocentesis is very important procedure which is done for detecting or for diagnosing genetic disorders. What is the impact of genetic disorders on a family or a patient? Genetic disorders impact not only affect the physical health but also the psychological, mental health and the social well-being, that is the social health of patients and their families. Understanding the unique aspect of genetic information and anticipating reaction to genetic tests and diagnosis can help guide a course of action to minimize distress and maximize benefit for both the patient and family. So if the family members have a little information or they get full information about the genetic disorders or what are the various tests, the prenatal diagnostic test or the prenatal screening test for genetic disorders, this can help to minimize their stress, and it will provide good benefit for the patient and the family. An increased genetic risk or a genetic diagnosis can substantially impact medical management as well as the psychological and social well-being of the patient and family. So like I said, if there's any genetic risk, so that can have an impact on the physical health, the mental health, social health as well as the medical management. The personal and permanent nature of genetic information raises a range of emotions including guilt, fear, and helplessness. Specialists such as genetic counselors, social workers, psychologists, as well as support groups, they can be very helpful to the patients and families and to deal with them during these difficult times. So, patients or family members, they may be having emotions like guilt, feeling of fear, fear of losing one's health, or being helplessness, okay, not able to do anything. So at this moment of time, genetic counseling is very important. Then couples with an affected child often face difficult family planning decisions because future children may also be at a higher risk. So it is very difficult for the couples also, okay, during this situation. And depending upon the condition, parents may also be faced with hard choices regarding prenatal testing and termination of pregnancy. So, termination of pregnancy also is very um, we can say it is as sinful. Okay, so if the parents or the couples they find out that that their fetus is having a that their baby is going to have a genetic disorder, so it is very hard for them to terminate the pregnancy. The financial burden of a chronic genetic condition can also lead to stress among family members. So, not only the physical health, the mental health, the social health of the family or the patients are affected, but also the financial. There will be financial crisis, okay, in terms of money. Okay, so that is also one of the impact of genetic disorders on family. So I hope you are clear with the topic of today, that is on genetic diseases. A little bit of introduction or a brief introduction about what are genes, what are DNA, and the impact of genetic disorders on family. So thank you for the class and have a good day.